Hello again, this is Jared, and this is the third time I've started recording this. Hopefully I get it right this time. Anyway, I'm going to be reading an article by Dr. Ray Peat, and this one's called Multiple Sclerosis and Other Hormone-Related Brain Syndromes, and it's from 1993. And this one is close to my heart as I have a friend potentially dealing with some type of neurodegenerative issue, and so that's kind of how I decide on what articles to read. A lot of the time is someone important in my life is dealing with that particular condition. And, I don't actually expect them to read this or listen to this, to be honest. But, we've all been in that situation where we want to share and we think that this, whatever, may help them if they were, I'm not a doctor. I'm not trying to advise or suggest anything. I'm just saying that we want to help our friends, and if they take it to their doctor and want to utilize something, we would hope that they would give it a chance, But anyway, I don't expect my friend to do that. I hope. I wish they would. I wish they do. But I hope that this is helpful to someone out there that is having this issue and is open to reading the data, the research here that Dr. Peat is bringing, before us. So, anyway, this is how it goes. Since I'm trying to discuss a complex matter in a single article, I have separately outlined the essential technical points of the argument in a section at the beginning. Then I explain how my ideas on the subject developed, and finally there is a glossary. If you start with short day brain stress, estrogen's effects, and symptoms and therapies, you will have the general picture and can use the other sections to fill in the technical details. So those are three sections below that if you wanted to skip through to, I have them marked in the description, so you can skip to them. Anyway, the first portion, though, is the argument, and I probably will not read the glossary. Okay. Number 1. The hormones pregnenolone, thyroid, and estrogen are involved in several ways with the changes that occur in multiple sclerosis, but no one talks about them. 2. The process of myelination is known to depend on the thyroid hormone. The myelinating cells are the oligodendroglya, and I'm probably saying that wrong. Practice beforehand, and anyway. In parentheses, we have oligodendrocytes, a little easier to say, which appear to stop functioning in MS, and sometimes to a milder degree in Alzheimer's disease and other conditions. The cell's absorption of thyroid hormone is influenced by dietary factors. Number 3, and he's referencing things with numbers here, so you can go down and read what he's referencing afterwards. The oligodendrocytes are steroid-producing cells, and steroidogenesis, I assume, is the creation of steroids. Steroidogenesis is dependent on thyroid hormone and on thyroid-dependent respiratory enzymes and on the heme enzyme P450-SEC, which are all sensitive to poisoning by carbon monoxide and cyanide. The steroid produced by the oligodendrocytes is pregnenolone, which is known to have a profound anti-stress action, and which appears to be the main brain protective steroid. So just there we have 3 references. So obviously highly referenced article he's creating here. Number 4. Lesions resembling those of MS can be produced experimentally by carbon monoxide or cyanide poisoning. The lesions tend to be associated with individual small blood vessels, which are likely to contain clots. Since all animals have enzymes to detoxify cyanide, the poison is apparently a universal problem and can originate in the bowel. Quote-unquote detoxified cyanide is still toxic to the thyroid. Pregnenolone and progesterone protect against nerve damage by the excitotoxic amino acids glutamic acid, aspartic acid, monosodium glutamate, aspartame, etc., while estrogen and cortisol are nerve-destroying acting through the excitotoxic amino acids. Excitotoxins destroy certain types of nerve, especially the dopaminergic and cholinergic types, leaving the noradrenergic types, paralleling the changes that occur in aging. The clustering of oligodendrocytes around deteriorating nerve cells could represent an adaptive attempt to provide pregnenolone to injured nerve cells. Every time I say oligodendrocytes, I feel a little bit of an accomplishment. Okay. Number 6. The involvement of hormones and environmental factors probably accounts for the intermittent progress of multiple sclerosis. To the extent that the environmental factors can be corrected, the disease can probably be controlled. All right. Not what you hear normally regarding these diseases. So, I hope that right there is just a little bit of hope if you're listening to this. All right. The first section that he referenced above, short day brain stress. Shortly after I moved from Mexico to Montana, one of my students, a 32-year-old woman, began having the same sensory symptoms her older sister had experienced at the same age, at the onset of multiple sclerosis. Vertigo and visual distortions of some sort made her consider withdrawing from the university. I'm not sure why she tried eating a whole can of tuna for lunch a couple of days after the onset of symptoms, but it seemed to alleviate the symptoms, and she stayed on a high-protein diet and never had a recurrence. She told me some of the lore of MS, that it mostly affects young adults between the ages of 20 and 40, that it is common in high latitudes and essentially unknown in the tropics, and that it is sometimes exacerbated by pregnancy and stress. Later, I learned that systemic lupus, erythematosus, and other quote-unquote autoimmune diseases also tend to occur mainly during the reproductive years. I discussed some of the implications of this in Bean Syndrome, and you can read that by Dr. Peat elsewhere. Having enjoyed the mild climate of Mexico, I became very conscious of the harm done to us by northern winters, and began developing the idea of winter sickness. In 1966 to 67, allergies, PMS, weight gain, colitis, and arthritis came to my attention as winter-related problems, and I assumed that the high latitude incidence of MS related to what I was seeing and experiencing. Studies in Leningrad began revealing the mitochondria are injured during darkness and repaired during daylight. I observed that hamsters' thymus glands shrank in the winter and regenerated in the summer. Shrinkage of the thymus gland is a classical feature of stress and usually reflects the dominance of cortisone, though estrogen and testosterone also cause it to shrink. Winter's darkness is stressful in a very fundamental way, and like any stress, it tends to suppress thyroid function. In the hypothyroid state, any estrogen which is produced tends to accumulate in the body because of liver sluggishness. I began to see that PMS could be controlled by certain things: extra light, supplements of sodium and magnesium, high-quality protein, and correction of deficiencies of thyroid and progesterone. In working on my dissertation, I saw that tissue hypoxia, or lower than optimal concentrations of oxygen in the blood, may result from estrogen excess, vitamin E deficiency, or aging. There is a close biological parallel between estrogen dominance and the other hypoxic states, such as stress/shock and aging. Section 2 here, that he referenced above, is called Estrogen's Effects. As a portrait painter, I had been very conscious of the blue aspect that can often be seen in the skin of young women. In pale areas, the color may actually be blue, and in areas with a rich supply of blood, such as the lips, the color is lavender during times of high estrogen influence, around ovulation and puberty, for example. During these times of estrogen dominance, the blood is not only poorly oxygenated, but it has other special properties, such as an increased tendency to clot. The Schutz work in the 1930s began with the use of vitamin E to antagonize estrogen's clot-promoting tendency and led them to the discovery that vitamin E can be very therapeutic in heart disease. More recently, it has been found that men with heart disease have abnormally high estrogen, that women using oral contraceptives have higher mortality from heart attacks, and that estrogen tends to promote spasm of blood vessels. These reactions are probably related to the physiology of menstruation, in which progesterone withdrawal causes spasms in the spiral arteries of the uterus, producing endometrial anoxia and cell death. In toxemia of late pregnancy, or eclampsia, the exaggerated clotting tendency caused by excess estrogen or by inadequately opposed estrogen, i.e. progesterone deficiency, can cause convulsions and strokes. Vascular spasms could be involved here too. The stasis caused by the vasospasm would facilitate clotting. Vascular spasm has been observed in epilepsy too. Epilepsy can be brought on by the premenstrual excess of estrogen, and in the situation there is no evidence that clotting is involved. Leakage of hemoglobin out of red cells can cause vasospasm, so bleeding, clotting, strokes, and seizures can interact complexly. The brains of women who have died following eclampsia show massive clotting in the blood vessels, and their livers are characteristically injured with clots. Tom Brewer and others have shown very clearly that malnutrition, especially protein deficiency, is the cause of toxemia of late pregnancy. In Nutrition for Women, I discuss the importance of protein in allowing the liver to eliminate estrogen. And if you guys have not read any of Dr. Brewer's work, Tom Brewer, as he's mentioned here, you can check him out at drbrewerpregnancydiet.com, I believe. But anyway, if you look that up in Google, you'll find it. Fantastic website. Some incredible articles. If I were you, I would go and read some of that if you are interested in this protein conversation, or if you're pregnant or wanting to become pregnant. Okay. Various researchers have demonstrated that the plaque of MS usually occur in the area served by a single blood vessel, and some have suggested that clotting is the cause. MS patients have been found to have abnormal clotting time, and it has been suggested that an altered diet might be able to correct the clotting tendency. Studies in animals have shown clearly that a protein deficiency increases the fibrinogen content of blood. And that's from Field and Dam, 1946. Other factors that increase blood clotting are elevated adrenaline and cortisone. Protein deficiency causes an adaptive decrease in thyroid function, which leads to a compensatory increase in adrenaline and cortisone. The combination of high estrogen with high adrenaline increases the tendency for both clots and spasms of the blood vessels, plural. Okay. In experimental poisoning of animals with carbon monoxide or cyanide, the brain lesions resembling MS include blood clots. The patchy distribution of these spots in the brain suggests that the clotting is secondary to metabolic damage in the brain. Presumably, the same would be true in ordinary MS, with clots and spasms being induced in certain areas by metabolic abnormalities in brain cells. The injured cells that are responsible for myelination of nerve fibers are steroid-forming cells. A failure to secrete their protective pregnenolone could cause a local spasm of a blood vessel. The circulatory problem would exacerbate the respiratory problem. Steroid production is dependent on NADH and NADPH, and so requires adequate energy supplies and energy metabolism. The phenomenon of blood sludging, studied by M. Knisley at the University of Chicago in the 1930s and 1940s, is apparently a general result of decreased energy metabolism and is likely to be a factor in energy and circulatory vicious circles. And then the final section Dr. Peat referenced up above is symptoms and therapies. Around 1976, I met a woman in her mid-30s who heard about my work with progesterone in animals. She had been disabled by a brain disease that resembled MS or Devix, I'm not sure how to pronounce that, D-E-V-I-C, Devix disease, inflammation of the optic nerves. It would sometimes cause blindness and paralysis that persisted for weeks at a time. During remissions, sometimes using a wheelchair, she would go to the medical school library to try to understand her condition. She came across Katarina Dalton's work with progesterone and convinced a physician to give her a trial injection. Although she had trouble finding people who were willing to give her progesterone, her recovery was so complete that she was able to climb stairs and drive her car, and she came to my endocrinology class and gave a very good and long lecture on progesterone therapy. Although her sensory and motor functions became normal, she remained very fat and chronically suffered from sore areas on her arms and legs that seemed to be abnormal blood vessels, possibly with phlebitis. She appeared to need thyroid hormone as well as larger amounts of progesterone, but never found a physician who would cooperate, as far as I know. In the late 1970s, I was seeing a lot of people who had puzzling health problems. In a period of 2 or 3 years, there were 5 people who had been diagnosed by neurologists as having multiple sclerosis. In talking to them, it seemed clear that they had multiple symptoms of hypothyroidism. They weren't severely disabled, since they weren't fat or lethargic. Their physicians hadn't thought they could be hypothyroid. When they tried taking a thyroid supplement, all of their symptoms disappeared, including those that had led to their MS diagnosis. One of the women went to her doctor to tell him that she felt perfectly healthy since taking thyroid. And he told her to stop taking it, because people who have MS need a lot of rest. And she wouldn't get enough rest if she was living in a normally active way. I can't help but laugh. Oof. The assumption seemed to be that the diagnosis was more important than the person. When I refer to a quote-unquote thyroid supplement, I mean one that contains some T3. Many people experience quote-unquote neurological symptoms when they take thyroxine, which is T4, by itself. Experimentally, it has been found to suppress brain respiration, probably by diluting the T3 that was already present in the brain tissue. Okay, this next section is called Metabolism of the Oligodendrocytes. The rate-regulating step in steroid synthesis involves the entry of cholesterol into the mitochondria, where the heme enzyme P450-SEC then removes the side chain of cholesterol by introducing oxygen atoms to produce pregnenolone. This enzyme can be poisoned by carbon monoxide or cyanide, and light can eliminate the poison. This could be one aspect of the winter sickness problem. Peripheral nerves are myelinated by essentially the same sort of cell that is called an oligodendrocyte in the brain, but outside the brain it is called a Schwann cell. It is easier to study the myelin sheath in peripheral nerves, and the electrical activity of a nerve is the most easily studied aspect of its physiology. Certain experiments seem to indicate a quote-unquote jumping, saltatory kind of deduction along the nerve between Schwann cells, and it was argued that the insulating function of the myelin sheath made this kind of conduction possible. This idea has become a standard item in physiology textbooks, and its familiarity leads many people to assume that the presence of myelin sheaths in the brain serves the same quote-unquote insulating function. For a long time, it has been known that heat production during nerve conduction reveals a more continuous mode of conduction that doesn't conform to the idea of an electrical current jumping around an insulator. Even if the myelin functioned primarily to produce saltatory conduction in peripheral nerves, it isn't clear how this process could function in the brain. I think of the issue of quote-unquote saltatory conduction of the nodes of Renvier as another of the fetish ideas that have served to obstruct progress in biology in the United States. A more realistic approach to nerve function can be found in Gilbert Ling's work. Ling has demonstrated in many ways that the ruling dogma of cell membrane function isn't coherently based on fact. He found that hormones such as progesterone regulate the energetic and structural stability of cells. Many people, unaware of his work, have felt that it was necessary to argue against the idea that there are anesthetic steroids with generalized protective functions, because of their commitment to a textbook dogma of quote-unquote cell membrane physiology. I think the myelinating cells do have relevance to nerve conduction, but I don't think they have serve primarily as electrical insulators. If the adrenal cortex were inside the heart, it would be obvious to ask whether its hormones aren't important for the heart's function. Since the oligodendrocytes are steroid synthesizers, it seems obvious to ask whether the production of pregnenolone in response to stress or fatigue isn't relevant to the conduction process of the nerves they surround. Old age. A biologist friend of mine who was about 85 became very senile. His wife started giving him thyroid, progesterone, DHEA, and pregnenolone, and within a few days, his mental clarity had returned. He continued to be mentally active until he was 89, when his wife interfered with his access to the hormones. In old age, the brain steroids fall to about 5% of their level in youth. Pregnenolone and DHEA improve memory in old rats and improve mood stability and mental clarity of old people. Pregnenolone's action is improving the sense of being able to cope with challenges probably reflects a quieting and coordinating of the quote-unquote sequencing apparatus of the forebrain, which is the area most sensitive to energy deprivation. This is the area that malfunctions in hyperactive and quote-unquote dyslexic children. Weakening of the sequencing and sorting processes probably explains the common old age inability to extract important sounds from environmental noise, creating a kind of quote-unquote confusion deafness. Insomnia, worry, and quote-unquote restless legs at bedtime are problems for many old people, and I think they are variations of the basic energy depletion problem. The oligodendrocytes were reported, and there's a study there, to be the source of the senile plaques or amyloid deposits of Alzheimer's disease. Hiroshi and Lee, who were just referenced in the above, showed the cells in different stages of degeneration, ending with translucent, quote-unquote, mucoid spots that stain the same as amyloid, the material in the senile plaques. This type of cell also appears to form a halo or crown around degenerating nerve cells, possibly in a protective reaction to provide the nerve cell with any pregnenolone the oligodendrocytes are able to make. The oligodendrocytes, the source of the brain steroids that people previously believed came from the adrenals and gonads and were just stored in the brain, myelinate nerve fibers under the influence of thyroid hormone. Thyroid is responsible for both myelination and hormone formation. In old age, glial cells become more numerous, and nerve cells become structurally and functionally abnormal, but usually there is no problem with the formation of myelin. In MS, the problem is just with myelination, and there are no senile plaques or defects in the nerve cells themselves. These differences suggest the possibility that Alzheimer's disease involves a specific premature loss of brain pregnenolone production, but not of thyroid. Recent work suggests a central role for pregnenolone and progesterone in the regulation of consciousness, and possibly in the brain's detoxifying system. Elsewhere, I have suggested that vitamin A deficiency might cause the excessive production of the quote-unquote amyloid protein. A vitamin A deficiency severely inhibits steroid synthesis. It is used so massively in steroid synthesis that a progesterone supplement can prevent the symptoms of vitamin A deficiency. I suspect that vitamin A is necessary for the side chain cleavage that converts cholesterol to pregnenolone. Iron-stimulated lipid peroxidation is known to block steroid formation, and vitamin A is very susceptible to destruction by iron in oxidation. Iron tends to accumulate in tissues with aging. Gajduzik has demonstrated that brain deterioration is associated with the retention of whatever metal happens to be abundant in that person's environment, not just with aluminum. One type of glial cell is known for its metal binding function, causing them to be called metallophils. According to Gajduzik, calcium and other di- and trivalent elements are deposited as hydroxyapatites in brain cells. in brain degeneration of the Alzheimer's type. Even early forms of Alzheimer's disease began at an age when the youth-associated steroids have begun to decline. If MS involves a deficiency of thyroid or of T3 within the oligodendrocytes where T3 normally can be made from thyroxine, many things including protein deficiency can block the conversion of T4 to T3. Those cells would necessarily be deficient in their ability to produce pregnenolone. But in young people, the brain would still be receiving a little pregnenolone, progesterone, and DHEA from the adrenals and gonads. This relatively abundant youthful supply of hormones would keep most of the body's organs in good condition, and could keep the bodies of the major brain cells from deteriorating. But if proper functioning of the nerve fibers requires that they be fed a relatively high concentration of pregnenolone from their immediately adjacent neighbors, with the amount increasing during stress and fatigue, then their function would be impaired when they had to depend on the hormones that arrived from the bloodstream. For many years it has been recognized the brain atrophy of Alzheimer's disease resembles the changes seen in the brain in many other situations. The traumatic dementia of boxers, toxic dementia, the slow virus diseases, exposure of the brain to X-rays, ordinary old age, and in people with Down syndrome who die around the age of 30. In menopause, certain nerve cells have lost their ability to regulate the ovaries because of prolonged exposure to estrogen. The cells that fail as a result of prolonged estrogen exposure aren't the same cells that fail from prolonged exposure to the glucocorticoids, but they have in common the factor of excitatory injury. Since people who experience premature menopause are known to be more likely than average to die prematurely, it is reasonable to view menopause as a model of the aging process. It is now well established that progesterone fails to be produced at the onset of menopause. The first missed period increases loss of calcium, symptoms such as hot flashes, etc., and that estrogen continues to be produced at monthly intervals for about 4 years. The essential question for aging in the present context is why the anesthetic steroids are no longer produced at a rate that allows them to protect tissues, including brain cells, from the excitotoxins. Using menopause as a model for aging, we can make the questions more answerable by asking why progesterone stops being produced. During stress, we are designed not to get pregnant, and the simplest aspect of this is that ACTH, besides stimulating the adrenals to produce stress-related hormones, inhibits the production of progesterone by the ovary. Other stress-induced factors such as increased prolactin and decreased thyroid also inhibit progesterone production. Stress eventually makes us more susceptible to stress. Menopause and other landmarks of aging simply represent upward reflections in the rate of aging curve. Individual variations in type of stress, hormonal response and diet, etc., probably govern the nature of the aging process in an individual. The amphetamine-like action of estrogen, which undoubtedly contributes to the general level of stress and excitotoxic abuse of nerve cells, is probably the only quote-unquote useful facet of estrogen treatment. But a little cocaine might achieve the same effect with no more harm, possibly less. The toxicity of catecholamines has been known for over 30 years, and estrogen-stimulating effects are partly the result of its conversion to catechoestrogens, which increase the activity of brain catecholamines. Estrogen's powerful ability to nullify learning seems never to be mentioned by the people who promote its use. The importance of a good balance of brain steroids for mood, attention, memory, and reasoning is starting to be recognized, but powerful economic forces militate against its general acceptance. Since the brain is the organ that can allow us to adapt without undergoing stress in the hormonal sense, it is very important to protect its flexibility and to keep its energy level high, so it can work in a relaxed way. It is the low energy cellular state that leads to the retention of calcium and iron, and to the production of age pigment and other changes that constitute the vicious circle of aging. And mental activity that challenges obsession and rigidity might be the most important brain energizer. Pseudo-optimism, humor as therapy, has a certain value, but a deeper optimism involves a willingness to assimilate new information and to change plans accordingly. All right. This next section is called supplements. There's 2 paragraphs here, and then we're on to the references. Nutritional supplements that might help to prevent or correct these brain syndromes include vitamin E and coconut oil, vitamin A, magnesium, sodium, thyroid, which includes T3. Sorry, I said T3, not 2T3. Okay, so one more time, thyroid, which includes T3. Large amounts of animal protein, especially eggs, sulfur, such as magnesium sulfate or flowers of sulfur, but not to take continuously because of sulfur's interference with copper absorption. Pregnenolone, progesterone if needed. Bright light, weak in the blue end of the spectrum and with protection against ultraviolet, activates respiratory metabolism and quenches free radicals. Raw carrot fiber and or laxatives if needed. Charcoal occasionally for gas or bowel irritation. Coconut oil serves several purposes. Its butyric acid is known to increase T3 uptake by glial cells. It has a general prothyroid action, for example by diluting and displacing antithyroid unsaturated oils. Its short and medium chain fatty acids sustain blood sugar and have anti-allergic actions, and it protects mitochondria against stress injury. P.S. So we have a postscript here. In 1979, a woman whose husband was suffering from advanced amyotrophic lateral sclerosis, or ALS, asked me if I had any ideas for slowing his decline. I described my suspicion that ALS involved defective metabolism or regulation of testosterone. In some tissues, testosterone is selectively concentrated to prevent atrophy, and ALS is a disease of middle age, when hormone regulation often becomes a special problem. In the late 1970s, there was discussion of a higher incidence of ALS in males, and especially in athletes. I told her about progesterone's general protective effects, its antagonism to testosterone, and its prevention of atrophy in various tissues. She decided to ask her doctor to try progesterone for her husband. Later, I learned that her husband had gone into a very rapid decline immediately after the injection and died within a week. The physician had given him testosterone, since, he said, testosterone and progesterone are both male hormones. Makes me want to shake him. Besides making me more aware of the problems patients have in communicating with physicians, this tended to reinforce my feeling that a hormone imbalance is involved in ALS. Although I haven't written much about testosterone's toxicity, Marion Diamond's work showed that prenatal testosterone is similar to prenatal estrogen in causing decreased thickness of the cortex of the brain. Both of these hormones oppose progesterone's brain-protecting and brain-promoting actions. And here are the 20 references from that article, and we have a glossary. That was a lot. I appreciate you guys listening, those that stayed with it. I hope this is helpful. And if you have any questions or comments, please leave them in the comment section. I would greatly appreciate it very much. And man, I hope this helps someone out there that is dealing with these problems. God bless you all, and see you next time.